Okay, this is the DNA RNA lab, and I just wanted to put a video on here to help explain because there have been some problems in the past. Just a brief description of what DNA and RNA is. DNA is made up of individual subunits called nucleotides, and the sequence of these nucleotides contain our genetic makeup. Every cell contains it, and every cell contains the blueprint for that organism. So the DNA lives in the nucleus, and that cannot leave the nucleus. So the way that we get the message out to the ribosomes where proteins are made, which is what the DNA genes code for, is a molecule called RNA, specifically messenger RNA. And so what happens is the RNA comes into the nucleus, transcribes the message, leaves the nucleus, and then that message is translated in the ribosome into a protein by another type of RNA. So the RNA that comes into the nucleus is called messenger RNA. It reads the DNA, then it leaves, goes to the ribosome, where it is translated by tRNA into proteins. So what you have to do in this lab is make these strands of DNA, messenger RNA, and then transfer RNA or tRNA. So what you need, and this is all on the website of course, I have given copies of the different nucleotides. So here's an example of a nucleotide, okay? The little white part down here is the phosphate. Nucleotides made up of three things: phosphate, a sugar, and a base. So the white thing is a phosphate. Every one of them has it, and it's the same. In the case of DNA, the sugar, which is color-coded black, is deoxyribose. Okay, so if you have a black sugar, then it is DNA. In this case, the base, which is yellow, is adenine, or abbreviated A. In this case, same thing, deoxyribose phosphate. In the green is guanine. I didn't cut these out, but blue would be cytosine, red would be thymine in DNA. Okay, again, black bases, black sugars, DNA. So if you can see the top of these, how they're cut out differently. So when you're going to have a strand of DNA, you can see the sugar attaches to the phosphate nicely like that, and then this would do the same thing that way, and on and on and on, in whatever sequence that it is, and I gave you the sequence on the lab. So the DNA strand that you're going to build is going to be like this based upon whatever letters, whatever sequence it is. So you need 2 adenines, 2 guanines, 2 cytosines, and 3 thymines. And again, that's on the lab, so don't worry if you didn't get that. Okay, so you're going to build this DNA strand this way. Okay, DNA only has thymine, RNA does not. RNA has a different base. So we talked about the RNA now. Here's the adenine with the RNA, but it's a different sugar. So the phosphate is still white, it's the same in both. The sugar is different. In RNA it is ribose, that's what the R stands for. In DNA, if you remember, it was deoxyribose, color-coded black. RNA, the ribose is color-coded brown. But the A is still the same shape and everything. So here we have cytosine, here we have guanine. So these three are all the same as far as the bases in RNA versus DNA. The difference is in RNA, you have the base uracil instead of thymine. Okay, so these things bind in very specific ways. If you have double-stranded DNA, should have cut this one out, but the guanine is going to bind with the cytosine this way. You see how that fits? The adenine is going to bind with the thymine that way. If you have double-stranded DNA, which is how it is. Now when transcription occurs, the DNA unwinds, and so that DNA strand in here, I'm just going to have 2 nucleotides together, okay, will be alone. Normally they're like this with other DNA. So it unwinds, so we have 1 strand, which is what I've given you. So what you need to do in the transcription part is to build the DNA strand that I gave you, and then do the corresponding messenger RNA strand. So let's do it this way. In DNA, if you have a G, the C is going to bind to it like this, okay? And then, we don't have enough hands. And then we have the A. In DNA, it would be a T here. But in RNA, we don't have any T's, so it's going to be the U. So that one would bind there. Let's see if I can do this. And you would continue on down the line, and they would bind like this. And so I think there are 6 bases that, or maybe it's 9, that I need you to show me. This looks like a ladder, okay? And so this is transcription. You should never, ever, ever, or I should never, ever, ever see anything that looks like this, because we have DNA and RNA in the same strand, and that doesn't happen, okay? So brown to brown, side by side, black to black, side by side, but then they would bind top to top as far as RNA and DNA. So that's transcription. Now with translation, what you're going to do is now take your messenger RNA strand that you just made, don't take it apart, but you remove the DNA, and then you're going to put RNA just like this on top of it. So in this case, the A would match with the U, and the C's and G's always go together. That's easy. So I know I don't have them cut out, but this is kind of how it would look, okay? And this is translation, and this occurs in the ribosome. So you're going to have two RNA strands, one messenger RNA that you got from hooking up with the DNA, and one transfer RNA. On those, you'll need 5 A's, 4 G's, 4 C's, and 5 U's total, okay? So I hope this helps. It's one of those deals where it seems like some students get it right away and some don't, but I hope the video helps you. But just remember, the DNA stays with DNA as far as side by side, okay? And then the messenger, or the RNA, will bind to it like that. Not that one though. Like that. C's always go to G's in anything. Okay, in DNA, A's and T's and A's match up. But in RNA, the T is replaced with a uracil, or a U. All right. I hope that didn't confuse you more, but if you have any questions, post them on the discussion board, and I'll hopefully be able to answer them. Good luck.